Tissue, formation and repair. Wound healing conditions required for wound healing systemic factors. These include good nutritional status and general health. Infection, impaired immunity, poor blood supply and systemic conditions, e.g. diabetes mellitus and cancer, reduce the rate of wound healing. Local factors. Local factors that facilitate wound healing include a good blood supply to provide oxygen and nutrients and remove waste products, and freedom from contamination by, e.g., microbes, foreign bodies, or toxic chemicals. Primary healing, healing by first intention. This type of healing follows minimal destruction of tissue when the damaged edges of a wound are in close apposition, e.g., a surgical incision. There are several overlapping stages in the repair process. Inflammation. In the first few hours, the cut surfaces become inflamed and blood clot and cell debris fill the gap between them. Phagocytes, including macrophages, and fibroblasts migrate into the blood clot. Phagocytes begin to remove the clot and cell debris, stimulating fibroblast activity. Fibroblasts secrete collagen fibers which begin to bind the wound margins together. Proliferation. Epithelial cells proliferate across the wound through the clot. The epidermis meets and grows upwards until full thickness is restored. The clot above the new tissue becomes the scab, which separates after 3 to 10 days. Granulation tissue, consisting of new capillary buds, phagocytes and fibroblasts, develops, invading the clot and restoring blood supply to the wound. Fibroblasts continue to secrete collagen fibers as the clot and any bacteria are removed by phagocytosis. Maturation. The granulation tissue is replaced by fibrous scar tissue. Rearrangement of collagen fibers occurs and the strength of the wound increases. In this time, the scar becomes less vascular, appearing after a few months as a fine line. The channels left when stitches are removed heal by the same process. Secondary healing, healing by second intention. This type of healing follows extensive tissue destruction or when the edges of a wound cannot be brought into apposition, as quii, varicose ulcers, and pressure, decubitus ulcers. The stages of secondary healing are the same as in primary healing. Healing time depends on effective removal of the cause and the size of the wound. Inflammation. This develops on the surface of the healthy tissue, and separation of necrotic tissue, slow, begins due mainly to the action of phagocytes in the inflammatory exudate proliferation. This begins as granulation tissue, consisting of capillary buds, phagocytes, and fibroblasts, develops at the base of the cavity. Granulation tissue grows towards the surface, probably stimulated by macrophages and a range of locally released chemicals. Phagocytes in the plentiful blood supply reduce or prevent infection of the wound. Some fibroblasts in the wound develop a limited ability to contract, reducing the size of the wound and healing time. When granulation tissue reaches the level of the dermis, epithelial cells at the edges proliferate and grow towards the center. Maturation. This occurs by fibrosis, in which scar tissue replaces granulation tissue, usually over several months, until the full thickness of the skin is restored. Fibrosis, scar formation. Fibrous tissue is formed during healing by secondary intention, e.g. following chronic inflammation, persistent ischemia, suppuration, or extensive trauma. The process begins with formation of granulation tissue. Then, over time, the inflammatory material is removed, leaving only the collagen fibers secreted by the fibroblasts. Fibrous tissue may have long-lasting damaging effects.